Assalamu alaikum, my dear students. Today we are going to start with the new thing in our lecture of cell injury and accumulations, and that is apoptosis. As in our previous lecture, I have told you that there are two types of the cell deaths. One is the necrosis that we have discussed in our previous lectures, and necrosis is a type of cell death that is a pathological one. Today we are going to discuss a physiological cell death, which is a procedure that is normally happening during the organogenesis, during the embryogenesis, and in the adult life, and also in the later on life, and that is called as apoptosis. So the learning objectives of our today's lecture will be that at the end of the lecture you would be able to define apoptosis with the examples. You would be able to differentiate between the pathological and the physiological cell deaths. You would be able to describe about the pathogenesis of the apoptosis, and the most important thing, that you would be able to draw extrinsic and the intrinsic pathways of the apoptosis. I want to stress upon two things because apoptosis is very important. From the apoptosis you are going to get almost two questions that are very important. One is about the differences, and the second one is about the extrinsic and the intrinsic pathways of the apoptosis. So if we are going to divide the cell death, I have just told you that we are having the pathological, which is necrotic type of cell death, and the physiological cell death. If we are going to divide the physiological cell death into further three classifications, that they are apoptosis, autophagy. Autophagy, my dear students, means auto means self, phagy means digestion. So if cell is going to eat itself by its own lysosomal enzymes, then it is going to be called as autophagy. And this is a very important phenomena. 2 years back, the Nobel laureate, which has got the Nobel Prize in the physiology, whose whole work is on the autophagy and its importance, and a bit of its research is also on the emphasis of the autophagy along with the fasting. And there are some other mechanisms, which is the composition of the apoptosis and the autophagy combined in the physiological cell death. If we are going to further divide the apoptosis, then it could be caspase dependent apoptosis and caspase independent apoptosis. My dear students, just remember this thing. Caspase is an enzyme and it is derived from C is for the cysteine and asp is for the aspartic acid. So in this enzyme we have cysteine and the aspartic acid, and there are basically two types of the caspases, intrinsic, which are going to initiate the intrinsic pathways, and these are the receptor caspases, and then mitochondrial caspases. One is the initiator caspases and the other one is the executionary caspases. So just remember, in caspases we have further classifications of its types of the executionary and the initiator. So initiator caspases are those caspases which are going to start the process of the apoptosis, while executionary caspases are going to end up the process of the apoptosis. And then we have another type of apoptosis, which is caspase independent, and we do not need to have a caspase for apoptosis to happen. For every cell there is a time to live and there is a time to die, just like a human body. So there are two ways in which cells are going to die. One, if they are going to be killed by an injurious agent in a pathological cell death, and the other way, if they are going to induce to commit suicide by themselves, or they know that aging has been done and our cycle has been completed, so we have to die. So this is a physiological cell death. So apoptosis, by definition, our first learning objective was here that you should be able to define the apoptosis, and the apoptosis is an appropriate programmed cell death. So basically apo means away from, okay, and tosis means to droop or to fall. So it's just like falls of the leaves. As you know that in autumn leaves are falling. So apoptosis has been derived from the same phenomena of the Greek word, that it's a falling of the leaves or falling away. So it's basically a programmed cell death, just like a falling of the leaves of the trees in the autumn. While necrosis, as I have already told you in a previous lecture, that it's a Greek word and it means deadness, mortification. So as far as the differences between apoptosis and necrosis are concerned, I have already told you that these are the differences, because the major difference is that apoptosis is a physiological one and there is no breakage in the cell membrane. If there is no breakage in the cell membrane, the cell contents are not going to leak outside, but they are going to be the formations of the apoptotic bodies, and then these apoptotic bodies are being engulfed by the macrophages and the other cells. So but in necrosis, we have already discussed all the types of the necrosis, and you have seen that after the blast formation, the cell membranes are going to rupture, and because of the rupturing of the cell membrane, cellular contents are going to leak outside the cell surfaces, and because of this we are having the inflammation outside these cells. So just remember this thing. And one thing that is very, very important is that basically apoptosis is a controlled autodigestion that I've just told you by the activation of the endogenous proteases, and these are one of the types is caspases that we have just discussed about the initiator and the executionary caspases. And as it is an autodigestion, which is a controlled one, so it results first in a cell shrinkage, membrane blabbing, nuclear condensation, and the formation of the apoptotic body. So the results, if we are going to see in a DNA fragmentation, that there is a DNA ladder formation. So here you can see that there is a ladder formation. That pattern is seen in a gel dog system. If you are going to do the PCR of the apoptotic cell, this is a ladder formation. And if you are going to see in a necrosis, what you are going to see, it's a continuous process, because of pathological ones there is all of the process is going to be involved. So the differences, if we are going to do apoptosis, results in a loss of the mitochondrial function, unlike necrosis. In apoptosis, the dying cell maintains its plasma membrane integrity. That is very, very important, I have just told you. And then the phagocytes are going to come, which are going to uptake the apoptotic bodies, and there is no inflammatory response. So this is your normal cell. This cell is going to receive the orders that it has fulfill its function, it has fulfilled its lifetime, so it has to die. So the apoptosis is going to start and cell is going to shrink, and if you can see, there is a condensation of the chromatin that is going on, and then because of the shrinkage there is a blabbing that is going to happen in this is the blabbing of the plasma membrane. After a few times, nucleus is going to collapse. Here you can see that there is a shrinkage nucleus, pycnotic nucleus, but now there is a karyorexis. Nucleus is also fragmented and nucleus is going to be collapsed, and there is a continuous blabbing, and if it is going to continue, there is a formation of the apoptotic body, and further there is a lysis of the apoptotic by the macrophages or the other cells. So So the differences is in necrosis we have a cellular swelling, while in apoptosis we have a cellular condensations. In necrosis membranes are going to be broken up, but in apoptosis membranes are going to remain intact. As I have already told you that necrosis there is a ATP depletion because of the less of the oxygen, the injury is going to happen. But what is going to happen in apoptosis, that it is because it is a process that is normally happening within the body, so it requires ATP. So in necrosis there is an inflammatory response alongside the cell, but in apoptosis there is no inflammatory response. If we are going to see a DNA fragmentation in the gel dog system, it is a random or a smeared one, just I have just showed you, while in apoptosis it's a ladder like, because every step is going to show in that system. In vivo, whole of the areas of the tissue is going to be affected, while in apoptosis individual cell is only involved. Whole of the area is not going to be affected, like you have seen in a coagulative necrosis, or we have seen in gangrenous necrosis, caseous necrosis. Whole of the tissue is not going to affect. It is only an individual cell that is going to be affected. So my dear students, as far as the importance of the apoptosis is concerned, there are two different reasons. Programmed cell death is basically needed for the proper development, and as in cases of mitosis, while the programmed cell death is needed to destroy the cells that are the threat for the integrity of the organisms. This point is very, very important. Why some cell is becoming a threat to the integrity of the organisms? I have told you in the hyperplasia and in the metaplasia that there is a rapid production in the hyperplasia of the cells, and cells are increasing in number because they are rapidly going into the cell cycle. There are chances that mutations can happen in these cells. So if there are the mutated cells because of the hyperplasia or because of the differentiation from one epithelium into another epithelium, like from a respiratory epithelium there is a change into the squamous epithelium, pseudostratified respiratory epithelium to the squamous epithelium, then there are chances that this squamous epithelium can have a malignant changes. So these cells become a threat to the integrity of the organism. So in a normal body, it is very, very important that these cells should die. So then what is the role of the body? That it's going to initiate the process of apoptosis. So importance in the embryogenesis, I have told you in the start of the lecture that it is needed basically to eliminate the cells. If you are going to see, these are your interdigital spaces, and in the interdigital spaces we have the membranes, and in order to remove these membranes apoptosis is needed, and then your formations of the toes is going to occur, and this is happening because of the apoptosis. Then elimination of the non-functioning cells. As you know, there are lots of cells which body are going to recognize that these are the non-functioning cells, so they should be eliminated. So this is going to happen by the process of apoptosis. Then you must have seen the process of the immunity in the thymus, which is we are going to call as self-recognition. The cell which are not going to recognize self-antigens, these cells, self-antigen recognizing cells, are going to be destroyed. So it is going to, because these cells can initiate the process of the rheumatic arthritis, SLE, which are going to damage their own cells in the body. So any cells that is going to be dangerous for cell's own self is going to be go under the process of apoptosis, and this is happening in immunity by the self-recognition of the self-antigen. Then organ size. This is very, very common. After the parturition in the women, after the delivery, the size of the uterus has to come back, and this can be achieved by the process of apoptosis. Then again a simple example that I've just told you. This is your virgin mammary gland, and in the pregnancy or the lactation, here you can see that the mammary gland has been increased in the size. Now after the lactation has stopped or involution in a non-pregnant or non-lactating, it has to go back to its normal size. So this is going to be achieved by the process of apoptosis. Then the second most important example is of the BPH, benign prostatic hypertrophy or hyperplasia, and this can be achieved with the help of the testosterone blocking drugs, and these testosterone blocking drugs are going to initiate the process of apoptosis, and then there will be the shrinkage of the prostate glands resultant from the previously increase in the size because of the BPH. Then the most important thing is, I have just told you, tissue remodeling, and this is going to happen. There are lots of cells when we are having any infections, so because of the infections, antigen is going to come within the body, and there are lots of cells that are going to form to combat this antigen or this infectious injury. Like in COVID, we have so many inflammatory cells that are going to be produced, and you must have heard about the cytokine storm, that there are so many cells that are going to be produced, inflammatory cells, but we don't need it. Once this antigen is removed, you don't need all of these cells to be present, because then these inflammatory cells can be self-damaging. So these are removed by the process of the apoptosis. So these are the few important things that we are going to discuss in the apoptosis. So cells lost by the apoptosis are replaced by the cell division. So this is an important balance that is always going on in the body. Just remember this thing. This balance is very, very important in order to maintain the integrity of the cellular organs and in order to maintain the numbers of the cells within the body. So this is an important thing. My dear students, thank you very much. We are going to discuss the other part of the apoptosis in our second lecture.